Welcome everyone to the second portion of our workshop.
It's harder.
We're going to get started now. For the next speaker, we have Michal Hammel, who is a research scientist at LBNL and a beamline scientist at Civils, and he will be talking to us about solution scattering.
Thank you for invitation again. My name is Michal Hammel, as I was introduced already. I'm a staff scientist at the Berkeley Lab, but also a beamline scientist with a single beamline. What we do there is a solution scattering, mostly on the biomolecules. So I will introduce you a little bit of the technique. Probably already expert in solution scattering on biomaterials, but it's good to just know the basics. And then I go straight to the modelings and AI and lots of AlphaFold and how we're using AlphaFold to get ensemble modeling. Small scattering is basic, very simple. We have samples in the solution, proteins, RNA, lipid particles, etc., etc. Incident beam, we have a light source, a scattering beam. We have scattering is super boring. In this case, it's just very symmetric signal on a detector. What we do with this, we have this our detector, and we averaging, spherical averaging, basically a signal and getting one curve. We subtracting buffer and sample, and this curve has the information. It's very minimal information, but with the combination with the atomic structures, we can do a lot, as you will see in a minute. Using this reciprocal space, we can translate it to the real space, and that's called pair distribution function. The pair distribution function have you some information about histogram distances of all the electrons in your biomolecule. And so you can get also some information about maximal dimension, overall shape, extension. This is really basic parameters. What we can extract from the curves is also quality of sample based on so-called linear plot, linearity, etc., etc. There is also information of molecular weight. And this is very important because before you start to model something with a solution scattering, you need to know your molecule is monodisperse, and don't try to model something what is 50 kilodalton with something what is 100 kilodalton, etc., etc. But it's very important, and it's also with the temperature melting points. What we have interesting talk before is defining unfolding. So we can quantitatively correlate these curves. It's a Kratky plot that is directly replotted from the scattering profile and from the shape, but also quantitatively we can identify how much unfolding we have in the protein, how much it change with mutations, with the ligands, etc., etc., etc. And this again, the PR function give you also some information about you have the domains, you have a maximal dimension where the PR function go to 0. So already in a curve, there's lots of information about structure on a low resolution. Just explain you that what is the histogram of the distances, where the PR function will go to 0. That's the maximal dimension of the protein, or whatever you have in V. And there's also some information. There's just overall shapes where the peaks are, etc., etc. Yeah, when the users come to the beamline or any kind of solution scattering, they are biologists and they don't really want to understand the technique. What they want is have some models, how the molecule looks like. Back in 1999, I was very young, so we started to do this ab initio modeling, and we developed some tools, obviously, also very big names. Svergen just continue and became kind of a guru of this ab initio modeling. It's interesting that from one-dimensional curve, this is PR function fitting, but we can do reciprocal space too. We can do some three-dimensional shapes, kind of ab initio beads. It's not unique solution because you go from 1 dimension to 3 dimensions, so you repeat it over and over, do some averaging, and get some idea how this looks like. What is much more important for this crew, especially 2021, we can now apply atomistic model. Especially, we're looking on AlphaFold model where the confidence of the parts are pretty high, but there are some bits they there are not really good confidence in. So now we can just start to move this molecule based on, I'll show you restraints from statistic coming from the AlphaFold especially. That's PLDT values and PAE matrices. And so that's what we're developing now. But before we do that, we need to have so-called a golden standard to come from the atomistic model to a scattering profile. And that's tricky, but we're getting there, and it's really getting better and better. So here's a just example how many details we can see in this curve. Again, curve, PRR functions. Here on the right side, you will see crystal structure of a small cell lace. And you see, oh, it's fitting pretty well. There's some discrepancies here in PRR function, some misfits here. And only what you're missing here in this case is a histone. The 7 residues they're sticking outside. So you can kind of now realize how beauty is that you can really add everything what you see in a solution. You can add to the model. You need to add to the model before you start doing any kind of movement. And the histone can be exposed in this case. It's lying here. Will be not really changing with it. So that's how many details you can start thinking about. What's going on? Okay, so I add this, how we're calculating the scattering profiles using the Debye formula. And the Debye formula with additional some kind of catch factor because proteins or any kind of have vibration layer that's good as well. So all that need to be also implemented is this fitting and getting the final intensity from excluded volume, hydration layer. So that is the math, but please don't be an expert. There's lots of literature about that, how you're going from atomic structure. And that's the beauty in comparing to the cryo-EM when you have blobs or densities. Here you have the real data, experimental curves that you can match with your models. Yeah, I'm a beamline scientist too, and since 2005, it's big mess. I know it's every beamline. That's right. It's not clean, but we have 2 technologies. One is high throughput. That's where take the sample batch, collect the data. That's this robotics here, going between plate and the beam that's coming here, fly to detector. We also have a pre-purification steps in automated mode 2. These 8 users can be collected overnight without any issues and multi-angle scattering here. So let's think about it, what we can do with the SEC SACs first, because that's relevant to the modeling. We want to have a clean model per sample. So we're collecting from the plate, we have some columns, they purify the aggregations, dimer, monomers, mixture, etc. 60 microliter concentration, about 2 to 10 minutes per node, 25 minutes per sample. We have SACs, of course, but we also have the multi-angle landscape. And that's additional bonus to you that you can calculate molecular weight not only from SACs, but also from moles. And you can compare, for example, in this case, compare across the elution, this is elution, can compare molecular weight, what is independent from the shape of the molecule, and you can compare to the radius of gyration, what is dependent on the shape of the molecule. And in this case, we see that it's very flat, so more of this per sample, the radius of gyration really change. And that's kind of why we see there is some thermodynamic equilibrium between open and closed state, and we can track it as it's eluting. So here is just a small molecule. Don't go to the biology now, but we need to have multi-state SACs model to explain the data. I'm not showing the data in this case. And we come up with this kind of final ensemble states of this complex, and this is just a movie for reviewers to see what in between the states, how far this complex can move. In high throughput mode, you know, SACs is a comparison technique, so you're comparing, but we can collect hundreds of samples in an hour. This is already high throughput for the ligand screening, especially for the companies that are interested in this. We do lots of high throughput screening for our vaccines, lipid formulations. It's just, again, we're not comparing and analyzing each data sets because you have so many of those. You're putting in this kind of so-called heat maps, comparing curve of A to B, C, D, and you're coming to the heat map. And it's a great example of a protein called AFA that was screened against hundreds of different compounds and find out different classes of compounds, how they change the allosteric of that molecule. Oh, this is important too. If you want to do SACs, obviously you have SSRL here. Thomas is right there, so don't contact me right now. But we do the mail-in Berkeley, that is light source. So we don't have a users at the beamline. They're sending us the samples in 96 Playwell or SEC SACs in Appendorf, and we collect for you. We analyze the data as well, and we give you the results. Obviously, come with the questions later. So some kind of information, how much sample, but everything is online. My homepage, our homepage, Sibyl's was there, but if you have a problem, type Sibyl's SACs. You will get to our information about the mail-in SACs. Just a little advertisement, but again, you got SSRL. This is how the robots for the high throughput works, just in times, going back and forth. It's nice to show that we really doing it in high throughput. Here on the right side, we have a needle as a sample as well. So needle have a transparent window for X-rays. And this is just a shots of as the needle is already in the post and the X-ray come through this way. You can kind of see size of the beam a little bit here. And on the bottom, you're just getting curves as it goes right there. So it is really high throughput. Again, this is high throughput in a batch mode. So recommend just comparison, different constructs, et cetera, et cetera. For the modeling, obviously the SEC. Another advertisements, but this is already related. So as you know, 2024, Google DeepMind Group and also David Baker got a Nobel Prize, leveraged the Sibyl's a lot because from 2015 till 2025, we collect about 4,000 different constructs for him. And we have about 30 papers, mostly some high-ranking papers. And all what he was doing, collecting the data and using our Fox application, it's the calculator of SACs, just matching with the constructs to see if the construct, what he designed, behave in a solution. So it's very high throughput. Let's go to AI, and I call it dynamicity. In 2018, 78, you've probably seen this already before. This is a slide borrowed from James Holton. He using to explain how important it is to do ensemble modeling on the side chains in crystallography, et cetera, et cetera. I just like to show it how important it is in a bigger proteins or bigger domain movement, et cetera, what we've seen before. The question was very simple. If the galloping horse have elevated legs all the time, and if you put this movie in a 1 sigma averaging structure, the most important part just gone. That's right. You don't see anything. That is important for questions like if the legs are moving. That's why this is just more relaxing slides. Let's go to the heaviness. AlphaFold prediction of one of the protein complex 3 chains colored with different colors. When we're putting in the PLDT colors, that's what a lot of people like to do, we kind of start to see the low confidence. But these are the colors based on PLDT, what is a local confidence of the fold. And you kind of can see where it's probably some flexibility unfolding, some kind of lower predictions values. And lots of people, when they do predictions, they don't looking on this, and that's pity, because this is very important information. It's called PAE matrix. It's a residue alignment confidence, and tell you how, for example, this residue is really confident in a position. If it's blue, it's very high confidence. It's darker. For example, this residue to this residue is already low. So I think this domain and this domain exists as a folded, but they may move relatively to each other. And for example, when you look at this and this, these two domains are really highly confident together, but the rest is kind of fuzzy. That's where you can start to think about it using this together with PLDT values, start to predict where the movement is happening and try to model it to the SACS data. And that's exactly what we do. We're using Leiden clustering to define the regions that are together, and they're supposed to be together and they're not together. Yeah, it's a graph-based community clustering algorithm. And we applied this clustering to this PAE matrix. So we not manually go and see where the domain seems to be moving or not. And again, this is a coloring based on a clustering algorithm of the same complex that I showed you on the slides before. What we do with this, we are AlphaFold predicted model. We can start to move it with a very simple molecular dynamics. This is not water box. This is very simple kind of coarse grain where we're heating up the parts with low PLDT values more. And the PLDT values there are really confident we're not heating up, and we're running this very simple vacuum-based. And we can organize this movement based on experimental radius duration coming from the experiment. So we can just also restrain this movement a different way based on a SACS data already. And so what we do, we apply a calculator and see the AlphaFold. This is this one. It's a poor fit. We define the fit based on chi-square. It's a goodness of fit. And we really need, in this case, when we have a one state, the fit is better, 5. But when we're adding more structures in it, that's called ensemble modeling, you can improve the chi-square to the specific value. And going to, I can tell you which value is the best because it's depending on signal to noise ratio. But you kind of need to get an expert involved with it. So we like to use these residuals to look at how good is the quality of fit. And this is just to show you how big we can go. This is one of the biggest protein in the human body. It's called DNA PKCS. And these are the Morph movie. You can play it again. It's too fast. Oops. Sorry for that. I just want to appreciate that the different AlphaFold model already predicted this kind of movement. So that's where the AlphaFold is already very useful. Just tell you comparing different predictions to see where we are. But none of those models, the single model fit the data well. The chi-square is 30. It's just very bad. And now again, we have this huge matrix. So let's use it. Define the regions that are more flexible, more rigid, and let's move the molecule in our MD simulations, calculate 10,000 different conformation in less than an hour. Again, this is not very computing time consuming process because it's a minimal MD. And fit the data. Obviously, you need to have the data. And what we see that the quality of fit with the 3 states or 2 states in this case is dramatically improved. And also explain biology. So there's also another papers that show that we really need to open in these heat domains. We call it these heat domains to really interact with other DNA repair factors. But we can see it now. And a data phosphorylation happen, forced to really opening these heat domains. So that's where it is useful for real application. And I like to talk about application rather than math. And we can go to details of how we do the PAE clustering in details, but you will appreciate what the SACS is very high throughput in this case. These are just example from just one our program. It's a DNA repair program. This is just a few of those. But you immediately see when the colors are, when we color the structures based on the clustering regions, you need to define the structure as ensemble. These are large movement. These are a little larger movement too, but it's kind of allosteric. So we're going deeper and deeper to more details. It's also depending the quality of data you get. You can do also some interestingly disordered proteins completely. You see the data already that is completely intrinsically disordered protein, but you can just fit it with a really high pipeline. We call it BilboMD pipeline. You can go online and just use it right now. But don't put everybody some SAX data. Hopefully you don't have SAX data. Anyway, it's just example how the AlphaFold models do not match data, where this is a chi-square logarithmic scale. These are the PTM values. It's just overall prediction quality of the model. And all of them need to be moved a little bit or more. This is just only 50 SAX data matched to the AlphaFold. We need to really use the BilboMD or any kind of ensemble modeling to optimize the solution scattering fit. Okay, let's go to the application. I saw there was some application from cryo-EM. It was really surprising to me that they use IgG antibody too as a defined ensemble modeling with cryo-EM. Here's just an example what we can do with the solution scheduling. So this is a regular kind of, could be AlphaFold prediction of some IgG4 in this case, from SARS-CoV-2. Back then, we did lots of SARS-CoV-2. From the PRL function, you already see some interaction with antigen. But I don't want to talk about this movement here of the PRL functions, where you see the binding of antigen. But you can also see which antibody interact with antigen and not based on one single value or just this movement in the PRL functions. So antibody looks like this. Obviously, it's very flexible. There's some glycosylation. We have the FAPS and the FC region in the bottom. And so we are running BilboMD pipeline. And we not all show that this is open and closed, but we can, based on these are different antibodies, based on distribution of frequency of open and closed. This just means like two states model. We can kind of see, based on this frequency of open and closed states, which antibody is flexible, which is less flexible, more rigid. That means how much FAPs are moving. Why it is important for especially drug discovery or biotech companies. Just one example. This is not SARS-CoV-2, anti-CD4. It's a cancer treatment target. And they find out that when you delocalize this anti-CD4, you get an increase in agonism. That's what you want. And they ask, what is going on? And just looking at the SAX data, you see a difference is again in the Kratky plot, but what I can see from here, there is a different opening and closing between delocalized and glycolized antibody. And I have another 5 minutes, that's right? Well, this is, I'm gonna skip this one. This is just more details about this. I'm gonna go straight to the RNA. We can do epitope mapping the same way. Predicting epitopes by AlphaFold is very difficult. There is no evolutionary trace of antibody interacting with antigen. So you're predicting different conformations of antigen and antibodies, and you can, based on the SAX data, you can find out which epitope is really the right one from these 4 or 5 different predictions. We are going to the RNA. RNA is a different beast because have much more plasticity. And so before we do some sampling and feeding to the data and selecting which RNA prediction is right, we can use deep fold RNA, Google DeepMind, RNA compositing, so lots of AI tools now. But before we go into feeding these different structures of RNA to the SAX data, you need to have a better calculator. One of the reasons is because the magnesium contributes to the scheduling much more than water. So we use the graph neural network in this case. We train the model based on 10,000 structures in PDB from RNA structures. And this works to just predict where the magnesium should be or could be. And then we feed the data with ensemble or single states. And it's really work just for distinguishing which predictor is right. You can AlphaFold, give you this structure, deep fold, give you this structure, and you don't know which one is right. But now when we have a great calculator, at least improved one, we can see that, and we optimize the flexibility based on kinetic, not MD simulation in this case. We can select that, okay, AlphaFold was wrong in this case. And it's known that AlphaFold is still not good in RNA prediction structures. There's different tools that I recommend so far. Maybe do Google DeepMind work on something. We did it on 20 different RNAs or something like that, and we find out it's really the flexibility is important part to optimize the fit. But adding the magnesium have a significant contribution to really get the good fit. If you want to use the BilboMD, super friendly, or Scooper, that's the RNA stuff. Sequence, experimental data, you get some kind of nice picture about the fit, improvement, some models, and also validation. If the model fits right, you're not the expert, but you can read some feedback, what you may improve, how you improve the clustering or movement. You can adjust that on our app. And this is the group. Most important, Greg and Scott and me started it 20 years ago. That's amazing, still exists. Younger generation are still there. And these are all the founders that we rely on. So we are not like LS founded. We have own independent funding that we try to hopefully have one or two questions.
Yeah, we had time for maybe a few questions. Yes.
Oh, thank you for the great talk. I was wondering, because you mentioned earlier that if when you added 3 states instead of 1 state, you saw that the chi-square was getting lower. I was wondering if this could be due to the noise level, like higher noise level attributed from the 3-state system, and that's why the chi-square is getting lowered. And if so, is there any way?
Sorry, that was my timer to stop the talk. That's a great question. So it's really depending on the quality of data, how much you can say. And the value, sometimes you have a great chi-square like 1, but it's super noisy. So you don't know if it's really true. But if you have a changes like a 5%, 1%, I just give you some numbers in a chi-square between 1 and 2 states, that's not significant. So or rather look, I mean, again, you need to have some empirical knowledge about looking the residuals of fit rather than the numbers only. But the numbers are indication. When you have a 2 and 1, obviously it's way better. So it's really a comparison. SACS is about comparison rather than just the number of the chi-square.
Yes, thank you. Yes.
When you say in bias and simulation with PLDT value,
Yeah.
then like how exactly they do it? They just add force and bias energy on force, then like how they determine the direction?
Yeah. So far now the BilboMD work on a threshold. Everything what is below 50, we say it's flexible. Everything what is above 50, it's not flexible. And also look at if it's a part of the cluster or not. So we take the cluster, we calculate the average B factor. We're working now to heat up each residues in a specific temperature. It's tricky based on a PLDT value. So instead of this movement domains, you will have a real plasticity where you can take each PLDT value of the residue and heat up in a specific way, which is lower PLDT, higher temperature, smaller PLDT, or opposite, you know what I mean. So that's what we work on, but it's a threshold 50. It's very, we're still doing something on it. So yeah, it's a great question.
So is there a way to, for RNA work,
Yeah.
Is there a way to kind of calculate theoretical RDC measurements based on your?
Yeah, so RDC is from NMR. Yeah, so we are just publishing now a paper with the proteins, 2 domains, and try to find out the RDC, how they're correlating with our ensemble models. You can calculate from the models RDCs for sure. it's good to compare to the experimental RDCs. That's what you want to do. The bottleneck, what we find out, it's almost published now, so I can talk about it. So the SAX is good in, not completely in like angles, but and more like movement in translational way. And that's where the SAX can contribute to tell you, NMR gives you the angle, but the SAX give you actually the flexibility and the breathing or just extension. So the combining those both is feasible to improve not only the RDC values, but that's what I don't know about RNA. If we can calculate the RDCs, I'm not expert in NMR, but I think it's possible. Yeah, I think so. Well, I don't know, sorry.
Thank you, Michal.
Yes.